Hello everyone, my name is David King and I'm currently a senior majoring in biochemistry, and I'll be presenting my project synthesis of a novel fluorescent molecular probe for the diagnosis of Alzheimer's disease. Alzheimer's disease is a detrimental and progressive neurodegenerative disease that usually affects people above the age of 65, and it becomes more prevalent in individuals above the age of 80, where it affects 1 in every 6 people. And it's a very common disease, unfortunately, as the symptoms manifest in memory loss. It's a very pervasive form of dementia, and many different symptoms can accompany, including even forgetting how to read or write. And misfolded proteins are the central role of why Alzheimer's is what basically causes Alzheimer's, right? So, as these proteins misfold, what happens is once one misfolds, it prompts another one to, and it's like a chain reaction, and it creates these plaques that are circled in white over here. And these plaques are what destroys our neurons. This can go on for, years and years and years, where most of the brain will be destroyed by these plaques. Traditional methods of diagnosing Alzheimer's disease, however, involve toxic radioactive materials and expensive and provides poor resolution. So the goal of this project is to synthesize a small molecule, which will be known as the fluorescent molecular probe, that can bind to these protein plaques, providing a safer and earlier diagnosis. Toxic effects can happen in the brain up to 10 years before a person is diagnosed with Alzheimer's, because symptoms usually do not show at that time. So providing an earlier diagnosis ensures the longevity of the patient and will also provide a more efficient diagnosis. And it will be characterized through NMR spectroscopy and elemental analysis, and it will be determined, the thermal stability and fluorescent properties will be determined through thermogravimetric analysis and UV vis spectroscopy, respectively. For the synthesis portion, the first step was an Arbuzov reaction dealing with this triethyl phosphite, which is a trivalent phosphorus, with this dihalide, and it basically adds on to either both sides of the ring, making this pentavalent phosphorus, which will be used in our Horner-Wadsworth-Emmons reaction. Our tert butoxide will generate a strongly nucleophilic ylide to attack this electrophilic aldehyde, aldehydic carbon, which will add these two pyridine rings to form our conjugated system, which will be our backbone for our final fluorescent molecular probe. This will be used in a alkylation reaction known as amine quaternization to form an organic salt. And this iodine counter ion over here can actually be exchanged to other counter ions, and this will potentially enhance the fluorescent properties and the carbon chain length modification to augment lipophilicity. We can, instead of using iodomethane over here, we can use iodoethane, iodopropane, and this will increase the hydrophobicity of our molecule, which will make it more um, which will increase the affinity of crossing the blood-brain barrier. And the blood-brain barrier is that selective barrier that makes sure that anything that's not supposed to go in the brain doesn't go. And we want to make sure that it crosses that selective barrier in order to bind to the plaques. And this is our meta C1 styropyridinium iodide. Our NMR spectrum over here shows our proton peaks. So over here, A is our most downfield, which is neighboring that um, quaternary amine and this vinylic hydrogen all the way over here. And our most upfield being our methyl peak that is directly attached to this nitrogen atom. Over here is my solubility properties for this meta C1 styropyridinium iodide. And over here, hexane, chloroform, and ethyl acetate, they are, hexane and chloroform are nonpolar solvents, while ethyl acetate's weakly polar, and methanol, water, and DMSO are highly polar. So for our fluorescent to show over here, that means that our compound has solubility in these polar solvents, while it does not have solubility in these nonpolar solvents. And for it to have water soluble character, that means that it's suitable for our brain because our brain is an aqueous medium, and it will be suitable for in vivo imaging for it to potentially bind to those protein plaques. And this is dilute concentrations for fluorescent measurement at 10 to the minus 6 molar, and it has very strong fluorescence at this very dilute concentration, which will provide improved Alzheimer's diagnosis because it can be used in many different microscopic methods for it to show that really high fluorescence. For example, fluorescence microscopy, for example. And then over here, figure 5 is our emission spectrum of the organic salt that was made. And for an emission max of 493 nanometers, this lies around the blue-green part of the visible spectrum. And we can see over here, it does have a blue-green fluorescence. It also has a white fluorescence. It cannot be seen here because the camera mostly picks up on the UV light, but since it's from under the curve from 400 to 700 nanometers, that's a mixture of red, green, and blue light, which will form white light. And that is a rare phenomenon in organic molecules to emit white light. And then over here is our TGA thermogram with a very high temperature stability at 276 degrees Celsius. And right after it melts at around 260 degrees Celsius, it will decompose, but it possesses high stability at this high temperature. And our UV visible absorption spectrum of the pyridinium iodide has a lambda max of 366 nanometers, and this tells us the wavelength where the maximum number of photons are absorbed. So to conclude, the final probe was synthesized, and the quantum yield for our final probe was 36.71%. So this tells us how efficient it is at emitting light, and 36% is regarded as a very high percentage for quantum yield, which is also known as quantum efficiency. So the higher the quantum yield, the better it is at emitting light, which will make this very good for microscopy. And we discussed earlier that it can undergo ion exchange reactions or carbon length modifications for it to um, be easily crossed through the blood-brain barrier, or for it to have enhanced fluorescence properties. And also, we want to do biological testing to make sure that this molecule can bind to these protein plaques for in vivo imaging. And this research project was made possible by a grant from the National Institute of General Medical Sciences from the NIH. Thank you very much for Nevada Embry. And if anyone has any questions, please let me know. And thank you for viewing this presentation.